Alright, so today's topic is all about the discovery of DNA, and our essential question is, can you explain the different scientists involved in the discovery of DNA? So when we learned about Gregor Mendel and his laws of inheritance, um, we have to understand that Gregor Mendel didn't know what DNA was. He didn't know what the actual molecule, what it was that was being passed off. It wasn't until a couple of these scientists that came along who did some studies and some research before we knew what DNA was and what it was made out of. So the first guy to dig into DNA was Frederick Griffith, and he wanted to know why some bacteria caused disease and why others didn't. There was some strains that caused the disease and some strains that didn't. So we had the disease-killing bacteria and the harmless bacteria, and he used mice as his source to study this. And he was able to figure out this process called transformation. And transformation is when one type of bacteria has been changed permanently to another. So something gets transformed into the non-disease bacteria to make it disease-causing. And this image down here shows kind of what he studied. So he had this disease-causing bacteria, the S strain, and to test it out, he put it in the mice, and sure enough, the mouse died of pneumonia. Then he had this harmless strain of bacteria, the R strain, and to test it out, he put it in a mouse, and boom, the mouse lived. Good for the mouse. So then he took this S strain, the one that caused the disease, and he heat killed it. He just put it to some intense heat, and the bacteria just became inactive. He put it into the mouse lives. That bacteria didn't cause disease. Well, then he was like, okay, what if I took a little bit of this S strain and a little bit of this R strain and mixed them together in a culture? Well, his guess was, since the disease-causing bacteria, the S strain, was no longer alive, that the mouse would live. But actually, lo and behold, what happened? The mouse died of pneumonia. So he figured out that something from this heat-killed bacteria, we don't know what it was, we don't know that it was DNA. There was something, though, inside that bacteria that got transformed into this harmless bacteria and made that bacteria harmful and made the mouse die. And later we'll figure out that that something that was inside it was actually DNA. Alright, so then this guy, Oswald Avery, came along in 1944 and he repeated what Griffith did. He and his team observed bacterial transformation, so he wanted to look more into, okay, what is transformation? What is being transformed? What is being passed along? And he wanted to find the particular molecules. He wanted to know what that was. So he discovered a nucleic acid, and we didn't really call it DNA yet, but he knew that it was a nucleic acid. And that nucleic acid is what transmits the genetic information from one generation of bacteria to the next. So he found out that that thing that was making the mice die, the thing that was being transformed from the harmful bacteria into the harmless bacteria, was a nucleic acid. And then we had Erwin Chargaff, a biochemist. So he wanted to study the chemical makeup of this nucleic acid. He wanted to study what is this made out of. So he found out that this nucleic acid that we now know as DNA is made out of a sugar, a phosphate, and 4 different bases: adenine, thymine, cytosine, and guanine. So, now we kind of know that this is DNA. This is deoxyribonucleic acid. This is what it's made out of. It's made out of a sugar, phosphate, and these 4 different nucleic acid base pairs. And he also discovered that adenine, the A, always pairs up with T, the thymine. And then that C always paired up with G, the guanine. So now we know that a nucleic acid is what gets passed off. And we now know that it's a DNA. We know what it's made out of. So then Alfred Hershey and Martha Chase came along, and they were experts in viruses. They looked at bacteriophages or bacteriophages. Those are special viruses that affect bacteria. So they looked at a certain bacteriophage that contained DNA and protein. So this bacteriophage had protein and it also had DNA in it. And so they wanted to discover, okay, is it the DNA that's getting passed along? Is the DNA inside of the organism? Is it outside the organism? Or is it this protein that's getting passed along? So they discovered, they did some experiments, and what they discovered is that it's the DNA, it's the genetic material. It's that nucleic acid that's being found in living things, and that's what's being passed along. So this image shows what they did. They had their bacteriophage, and they injected a radioactive material in it. And radiobacteria, the phosphorus 32, ended up getting passed along and found inside the bacteria. So we know that this virus passed along the radioactivity from its DNA into the bacterium. Well, then they wanted to see, okay, well, what maybe it's the protein that gets passed along. So they put radioactive material on the protein coat on the outside of the bacteria and had it infect the bacterium. And we found out that no, that didn't get passed along. So we know it's the stuff inside, it's the DNA that actually gets passed along. All right, so we know it's a nucleic acid. We know that's found inside. We know that that is what gets passed along to generations, or it gets passed along in viruses and bacteria. But what the heck does it look like? Well, this awesome woman, Rosalind Franklin, my gal, she was the one who really hit the ball out of the park with this. So she was an expert in X-ray diffraction, which is basically a fancy way of saying taking a picture with an X-ray light. So she used X-ray light that moved through the DNA, and she was able to take a picture. And this is kind of her little setup here. This is her setup. She had an X-ray tube that shined X-ray light out, and then a LED screen which filtered all other light except for the X-ray, so just X-ray lights coming through. And then it hits this crystalline prism, this glass prism, and inside that prism is a little strand of DNA. She was able to extract the DNA and put it in there. And then that prism diffracts the light. Right? We've kind of seen images of prisms diffracting light before, and what happens is that it kind of magnifies that DNA picture. So whatever got hit with light showed up on this photographic plate. And then the image that was formed was this right here. Aha. So now we're starting to figure out what DNA looks like. So this image really kind of defined the research of DNA going on in the time. This really, really was the catalyst to really figuring out DNA. So Watson and Crick then kind of, they kind of stole Rosalind's work and got credit for it in a way. But that's a different story. So with the help of Rosalind, with the help of her X-ray diffraction pictures, they discovered the structure of DNA. They discovered that it was in a shape called the double helix. And so they figured out that the sugar and phosphate backbone was in the DNA, so that was the backbone of the DNA, and then the nucleic acids inside of it. So now we know everything about DNA. We know the steps and the discoveries and the important people that discovered what it's made out of, that it's the thing that passes on our genes, and how it is formed, what it's shaped. So we know that there are three roles or functions of DNA: to store information, to copy information, and to transmit that information. So DNA stores all the information of how to be a cell, pretty much, how to function. And then that stuff is copied, and then it is transmitted. So it is copied and then passed on to the new cells from cell division.